Hello everyone, and welcome to today's live webinar, Genome Editing for Functional Genomics and Drug Discovery, presented by Chetana Revankar, a Senior Staff Scientist at Cell Biology R&D at Thermo Fisher Scientific, and Sanjay Kumar, a Scientist at Thermo Fisher Scientific. I'm Susie Valdez, and I'll be your moderator for this educational webinar, presented by LabRoots and sponsored by Thermo Fisher Scientific. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located at the lower left of the presentation window, type your questions into the box that appear on the screen, and we'll answer as many questions as we have time for at the end of the presentation. Also, notice that you're viewing the presentation in a slide window. To enlarge that window, just click on the screen icon located on the lower right. If you have any trouble seeing or hearing the presentation, click on the support button at the top right of the presentation window or use that Q&A button and let us know you're having a problem. I'd now like to introduce today's speakers, Chetana Revankar and Sanjay Kumar. Beginning with Chetana Revankar, Chetana is a Senior Staff Scientist at Thermo Fisher Scientific with over 15 years of experience in the field of cell biology and has been with the company for 10 years. Before joining the services team, she led the development of several new products ranging from cell-based assays for GPCR BacMam-based assays to 3D cellular models. As a technical lead on services teams, she supported project designs and execution. Her experience covers a broad range of applications from cell engineering to disease modeling. Our second speaker today is Sanjay Kumar. Previous to his current tenure as a scientist in the synthetic biology business of Thermo Fisher Scientific, Sanjay Kumar accumulated more than 10 years of experience studying the cellular response to DNA damage. That work began with Sanjay's pursuit of his doctorate degree, resulting in his PhD from the Cancer Genetics Department at Roswell Park Cancer Institute in Buffalo, New York. At Roswell, Sanjay identified the mechanism of replication fork slowdown upon DNA damage in fission yeast. He worked in the DNA damage field, continued with postdoctoral research at Caltech in Pasadena, California, where he dissected the role of the RIF1 protein in the vertebrate intra-S-phase checkpoint. Additionally, Sanjay holds a business degree in Bioscience Management with the Keck Graduate Institute. As part of the scientific team at Thermo Fisher Scientific, he has contributed to the commercial development of genome editing tools such as TALENs and CRISPRs. Most recently, he played a key role in the development of the GeneArt CRISPR nucleus mRNA and supporting early access to GeneArt Platinum Cas9, as well as TrueCut Cas9 V2. Currently, he's a leader of Cell Engineering Group within Customs Services team. His role requires Sanjay to lead several custom cell engineering projects and adapt these workflows for a variety of applications. Please join me now in welcoming Chetana Revankar and Sanjay Kumar. I will now turn the presentation over to them. Welcome to both of you.
Thank you. Hello, and welcome to the Genome Editing for Functional Genomics and Drug Discovery webinar. Our webinar today is divided into two parts. First part will be focused on genome editing to generate suitable cellular models, and the second part will be using these cellular models to ask biological questions. Without further ado, let's jump in. For the first part, we'll be looking at using genome editing tools, primarily CRISPR, to generate suitable cellular models. Genome editing workflow is a simple three-step workflow which can be very simply summarized as the 3 Ds: design, deliver, and detect. For designing portion, one needs to identify what gene they want to target, identify the correct target sites, and then design appropriate editing tool. For delivery, of course, once we have designed the tool, we need to deliver it, and we need to deliver it with the best possible efficiency. Once design and delivery are completed, we need to understand the efficiency of genome editing tool that was delivered into the cells, its efficiency, and the type of modifications that were made into the genome by this tool. And that's the detect portion. I will be going over design and deliver portions rather rapidly, but there are full on-demand webinars available on these topics at the Thermo Fisher website. You're very much welcome to go and check those out. To take the complications and all the work out of design portion, Thermo Fisher has generated a full library of pre-designed CRISPRs against all the coding genes in human and mouse genome. So how did we do that? We basically identified all the coding genes in human and mouse genome, and then identified which transcripts, which CRISPRs will be able to knock out all the transcripts of a particular gene. Identified all those CRISPRs, and then these CRISPRs were filtered and scored through several different criteria, which involve their GC content, presence of a protospacer adjacent motif or PAM, which is usually NGG sequence, then sequence preference, nucleotide positioning, and several other criteria, plus a lot of bench experience that we have generated went into making the scoring criteria that were used to make these database. After going through all of that, we went and looked at the top scoring guide RNAs, and those are presented in our online tool. With that, let's move on to the next step, deliver. Alright, so we have identified which gene we want to target and which format of CRISPR we want to use. Then we deliver it. Currently, the best format for CRISPRs is the Cas9 RNP format, which is a guide RNA, either in vitro transcribed or synthetic version, coupled to the true-cut Cas9v2 protein as a ribonucleoprotein complex, can be delivered into cells to achieve maximum editing. The Cas9 RNP can be delivered into cells using two methods. First is an electroporation based method using the Invitrogen's neon transfection system. In case you don't have that easily available, you can also use the lipofectamine CRISPR-Max reagent, and during this talk today we will be using CRISPR-Max reagent during our case study. In cases where you have a large library of CRISPRs to screen, in those situations lentiviral methods come in quite handy. And in the later part of the talk, we will be looking at use of lenti-array Cas9 coupled with lenti-array CRISPRs for a large-scale screen, and we will get there later. For now, let's just look at what's the next step. Next step is detection. So we have designed and delivered the CRISPRs, delivered them into the cells, and then how do we detect them? There are two simple ways to detect the editing outcomes. One is a gel-based mismatch detection assay, which I will be going through, and second is the DNA sequencing based method. After going through these two methods, we will be looking into a case study of generating a real cell line through transfection, clonal isolation, on and off target analysis. Alright, so we have made a CRISPR, and for every situation we usually recommend that a user design at least 3 CRISPRs for a given target. Test them to identify the one with the highest efficiency at the target site. Once that is done, we need to validate the edits in the pool of transfected cells and ask relevant biological questions, whether the modification was homozygous or heterozygous, which one was desired, whether we got any insertions, deletions, did they result in frame shifts, and if we were looking for any donor DNA based replacements, did we actually achieve homology-directed repair. So in order to address all of these questions, we go to the detect portion. So first is the gel-based mismatch detection assay. It's also called the genomic cleavage detection assay. It's a very simple, only 4-hour workflow from start to finish to address the question whether there was any genomic editing in the transfected pool. So, as we know, delivery of a genome editing tool into the cells will lead to genomic cleavage, shown here by scissors. So DNA double-strand breaks caused by the genome editing tool usually get repaired by one of the many pathways. The most predominant one is the non-homologous end joining. Non-homologous end joining pathway causes generation of small insertions or deletions in the DNA upon repair. And that's the insertion and deletion that this method will ultimately detect. How? Okay, so we have transfected the cells with the CRISPR. CRISPR generated double-strand breaks, and those double-strand breaks were repaired through NHEJ. What we do is then we harvest the cells, lyse them using the lysis buffer provided with the GCD kit, and in this case, you can see some of the DNA molecules contain some mismatched indels that are highlighted as red. Now this genomic DNA can be PCR amplified using the locus-specific primers, which will amplify wild-type DNA which was not modified, as well as the DNA that actually underwent some indel formation. Following this step, we can carry this DNA, or rather the PCR product, through a simple denaturation and renaturation cycle. And this denaturation-renaturation cycle, because of its random nature, will generate situations where one strand that contains an indel might get annealed with a different strand which may contain no indels, thereby making for these mismatched bubbles or mismatched heteroduplexes. This mismatched heteroduplex formation is very nicely detected by our detection enzyme mix provided in the kit, and the detection enzyme makes a cleavage at the heteroduplex, leading to the formation of 2 cleaved DNA molecules, which can be easily visualized by running the total product onto the gel, shown here in the gel schematically as a cleaved band which runs faster and an uncut band, that's the parental band. We can have an estimate of the total abundance of cleaved products, and that can give us a measure of the total amount of cleavage that was generated by the CRISPR. This method is very simple and easy. As I said, it takes only 4 hours to carry through. However, it's clear that it does not give us all the answers. To get more answers, we need to resort to sequencing-based methods. And I will go through the simple workflow using next-generation sequencing to address these questions. Once again, in this workflow, first step is delivery of the CRISPRs to cells, followed by transfection, and generation of the transfected cell pool. Transfected cell pool can then be subjected to PCR amplification as before. And after PCR amplification, we can generate a library of transfected DNA. This library can be taken through the usual sample preparation steps required for next generation sequencing, and then the prepared sample can be subjected to next generation sequencing using the ion PGM system, followed by sequence analysis. Here, I'm just giving you a snapshot of several thousand reads that are generated, but a simple snapshot can easily tell us that there are several different insertion and deletion events that have happened. All the deletions are visible pretty clearly, and I'm highlighting the PAM sequence, which is AGG sequence here, and the cleavage site would be somewhere over here. And you can see there are numerous insertion deletion events that are centered around the cut site. Getting an understanding of the frequency of these events can guide us in our downstream work. With this information in hand, let's go through a case study. First step in the case study is, of course, after assuming that we have figured out which gene to target and we designed all the CRISPRs for that target, is first step will be transfection. So let's transfect these cells with the CRISPR-Cas9, and we used, in this case, lipofectamine CRISPR Max reagent to generate DNA double-strand breaks, leading to the non-homologous end joining mediated indels. We took these transfected cells and subjected them to genomic cleavage detection assay and quantified the cleaved products, and by quantification we identified the CRISPR that I'm highlighting as EX5.2 CRISPR as the best performing CRISPR. So we decided to pursue this further, took the transfected cell pool and made the lysate, and used the lysate for on and off target NGS analysis to understand whether this CRISPR caused any spurious off-target cleavage events. In this case, there were no off-target cleavage events detected. Okay, so we have generated the cell pool and detected the best cleavage creating CRISPR. Next is using this cell pool derived from this CRISPR for generation of a real stable cell line containing knockout events for gene of interest. First step is using this cell pool, diluting it, and performing limiting dilution cloning or FACS sorting to plate one or less cell per well in a 96 well plate. After we do that, we can just leave the plates alone in a tissue culture incubator for a couple of weeks. After a couple of weeks, we need to look at these plates under microscope or visualize them through many other means to see whether the colonies are being formed. And a thing to pay attention is to ensure that we are identifying wells which contain only one growing colony. I'm showing here the example of two different wells. One of them has only one colony and the other one has two growing colonies. We want a single monoclonal population, so we should be going with the wells that have only one single growing colony. So we let them grow, consolidate them together, and go forward. So next step is a simple, easy Sanger sequencing based screen to identify clones whose DNA has some kind of indel events which are easily visualized on a Sanger sequencing chromatogram by degeneration of those peaks, which I am highlighting here. Candidate clone has several peaks that are degenerated and overlapping, whereas the wild type shows nice chromatogram. So these candidate clones we can pursue further and identify what are the indel events that occurred within. In this particular situation, we did sequencing of 178 single clones and identified 6 potential clones to pursue further using NGS. And in the next generation sequencing, we identified, and I'm showing you example of one particular clone. This clone had a 50-50 read distribution. So half reads arising from one allele, rest half arising from another allele, with 1 base deletion and a 14 base deletion. Both of these deletion events lead to a frame shift in the reading frame of the gene. Wonderful. So this is a very good positive clone that we can pursue further. Now you'll ask a question, okay, this is good for knockout. What if we want to just correct some sequence? So that is achieved by using a suitably designed donor molecule. Design of donor molecule is a different topic which we are not going to be touching here, but there are webinars on Thermo Fisher website which are available on demand, and we can look at those to understand the best ways to design donor molecules. Needless to say, we designed a donor molecule, and our goal was to convert an ATG to GTG in the coding sequence of a gene. And I'm showing you here two parallel examples of 2 different clones, one in which the ATG was completely converted to GTG across all reads, and in another situation where the ATG was converted only partly to GTG, and there were some reads that were still left with ATG, and the fraction of reads that contained ATG were half, GTG were half, so it looked like it was a heterozygous change. Okay, now after this detour, we can go back to our original case study. So we performed the limiting dilution cloning here, identified single growing colonies. Single growing colonies were scored through a simple Sanger sequencing method for presence of indels. The candidates that had indels were scored further and analyzed by next generation sequencing for the nature of indels. And in our case, I just showed you the example of a single clone which contained a 1 base deletion and a 14 base deletion. Wonderful. And this particular clone is ready for further analysis. Further analysis can be genotypic and phenotypic as well as functional. I will just go over the genotypic analysis where we understood where the deletion was, identified the junction points for the deletion, which I'm highlighting here. So between the red highlighted A and C was 1 base deletion, and that resulting frame shift led to formation of a stop codon, 17 amino acids downstream. In the other allele where we had 14 base deletion, again between these red highlighted A and C's were the 14 bases that got deleted. And let me point out, in both of these cases, the junctions of deletions are A and C sequence, but that's just a coincidence. It doesn't always happen. So in the second allele, between the A and C were 14 bases that got deleted. Resulting frame shift led to a stop codon 2 amino acids downstream. So we generated a stop codon here. Both the alleles of the gene have been converted to encounter premature stop codons and therefore lose their function pretty quickly. With that, I'm at the point where I can tell you about all the various cell engineering services that my group provides here. No cell line is too difficult for us. We perform a transfection optimization to identify the best conditions to deliver genome editing reagents into the cells. Then we can carry out a copy number assay to understand the copy number of the gene, because the cell lines are generally heterogeneous, so we need to understand that. We can also provide services for on and off target analysis, sequencing of the target locus to confirm its identity to reference genome that is present for human and mouse. And finally, we provide a complete end-to-end cell engineering service to generate the mutation or the gene knockout that you desire in the cell line of your choice. With that, we are ready to ask biological questions using these mutations that we just made. So I will pass over the baton to Chetana, and Chetana can walk us through the functional aspects of these mutations. How can we use these cellular models for asking relevant biological questions? So here is it to you, Chetana.
Thank you, Sanjay. Hello, everyone. So once you create these cellular models, let's see how you can use these to address questions about functional genomic screening or target identification. So in the next slide here, I want to ground everybody as to why CRISPR-Cas9 technology is so exciting for, especially for screening purposes. So in reference to what's currently widely popular, the RNAi-mediated gene knockdown, CRISPR-Cas9 technology is the next generation tool for functional genomic screening. And as opposed to the temporary gene knockdown, CRISPR-Cas9 technology provides complete and permanent knockout of gene expression, leading to stronger phenotypes, larger assay window, higher confidence in hits, and clear verification of the targets. Let's take a moment to see what basically functional genomics is and target identification. So the goal of functional genomics is to understand the relationship between an organism's genome and its phenotype, whereas target identification is the process of identifying the direct molecular target, or be it a protein or nucleic acid of a small molecule or a specific phenotype. So essentially, functional genomics is done on a larger scale or in high throughput mode. And why is functional genomics or target identification important? As Sanjay talked, making disease models from patient-derived cells or even regular cell line, has gotten so much easier with advances in cellular engineering or genome engineering. You can use these cellular models in multiple assay formats depending on your interest, and pick a pathway of your interest and try to identify key genes or targets in this pathway of interest and use these in combination, especially for combination therapy, to identify novel drug molecules or use the existing small molecules or drugs for therapy purposes. And CRISPR-Cas9 system allows us to do that by specifically knocking out genes of interest in a particular pathway or cell signaling pathway. So in my half of the talk, I'm going to focus on three different things. One is I'll talk about how phenotypic assays can be used to further investigate a gene knockout model. Then I'll talk about in vitrogen lentiviral-based CRISPR libraries for functional genomic screening. And then finally end my talk with how the lentiviral CRISPR library, especially the kinome library, was used to identify a potential new therapeutic target for rhabdoid tumors. For the phenotypic profiling of the gene knockout, the model system used in this case was ATG5 knockout cell line in U2S cell background, and it's shown here on this slide. This particular highlighted area shows that we picked one of the clones for ATG5 knockout, which was a homozygous knockout. It's represented by clone 3 here, and you can see the ATG5 expression is completely abolished in this case, and the housekeeping gene shown in green, the GAPDH, is expressed. And these studies were done using the Genomics cell and site high content imaging system. And before I get into the further phenotypic assays, let's see what role ATG5, or it's the autophagy 5 protein, plays in autophagy. So it's shown here in the complex autophagy process, and it plays a very important or central role in autophagy, especially formation of autophagosomes. And two other proteins, LC3B and P63, are also important in the autophagosome formation, and that's shown in this slide here, wherein we are looking to see what effect the ATG5 knockout has on the autophagosome formation. And as seen in this particular plot here, we are looking at LC3B spot intensity as plotted on the y-axis, and with increasing concentration of chloroquine, you can see in parental or the scrambled guide RNA, which is like the non-specific RNA treated cells, you see good LC3B accumulation in cells as determined by the spot intensity, whereas in case of the ATG5 knockout, we do not see the increase in LC3B intensity, indicating that LC3B does not aggregate in these cells. Similarly, in the next step to autophagosome formation is the autolysosome formation, wherein the autophagosome integrates with lysosomes, and one of the lysosomal marker is LAMP1, and you can see on the y-axis LC3B and LAMP1 co-localization is plotted. And in case of parental, you see good co-localization of LC3B and LAMP1 as shown by the red line. And in ATG5 knockout, you do not see this co-localization, indicating the autophagosomes do not merge with the lysosomes. The next, this particular image shows what I just described. Like in case of in the lower panel where you see the ATG5 knockout cells, you do not see LC3B aggregation upon chloroquine treatment, whereas in case of parental you do see. And then there is very little co-localization with LAMP1. Next, we look at the other important molecule in autophagosome formation, P62, which is, in fact, it serves as a receptor to target the ubiquitinated cargo to the autophagosome. And in case of the ATG5 knockout, as seen here, what you see is even before even before treatment with chloroquine, you do see increased levels of P62, and that is because it does not get degraded because the cycling or trafficking to lysosomes is defective in this case. So it just accumulates in the cytoplasm, basically. Whereas in case of parental, it does get degraded. And upon chloroquine treatment, you do see the puncta. So essentially in ATG5 knockout cells, the autophagosome formation is defective. The next thing we looked at is in these cells is the normal cytosolic stress. How do these cells respond to cytosolic stress? As we know that autophagy is basically a pro-survival mechanism. So are these cells more prone to cytotoxic stress? And if you look at this particular plot here, wherein we looked at caspase activation using the cell event caspase 3,7 reagent, there is, with increasing concentration of staurosporine treatment, there is no additional, or there is no, the ATG5 knockout cells are not really extra sensitive to caspase or to apoptosis, so indicating that these do the pathway the pathway for cytotoxic stress is not affected in this case. And on the plot on the right hand side, we looked at DNA damage using etoposide, and you can see with increasing concentration of etoposide, we do not see increase in DNA damage, or the DNA damage is comparable to that seen in the parental or wild type cells. And this DNA damage was detected using the phosphorylated H2AX antibody. And the same thing is shown in the images below, wherein you can see with 100 micromolar etoposide treatment, the activation or the levels of PH2AX are comparable in both cell types, ATG knockout as well as parental. This indicates that basically ATG5 knockout cells it just have defect in the autophagosome formation. So this is one of the ways wherein how you can investigate the phenotypic effect of a gene knockout. Next, I will move on to talking about the lentiviral-based CRISPR libraries for functional genomic screening. And the lentiviral CRISPR libraries come in two different formats. One is the lenti-array CRISPR library. As the name suggests, these are basically arrayed in 96-well format, and up to 80 genes or 80 targets are provided on one plate. Whereas the lenti-pooled CRISPR libraries are basically pooled libraries. And in this case, in the lenti-pooled library, each target has 10 guide RNA. And in case of the lenti-array library, each target has 4 guide RNA, and these 4 guide RNAs are pooled in the single well. So given the lenti-array format, because these are arrayed and you already know the identity of the target in the single well, you can use these libraries for plate-based assays or high-content assays. Whereas the pool library is mainly used for cell survival assays, and there is less upfront work for the pool library, but a lot of back-end work involving deep sequencing for when you use the pool library. Whereas for the lenti-array, it's less back-end work, as you already know the identities of the target. So in our catalog, we have 17 sub-libraries based on gene families and functional biology. And in addition to these 17 sub-libraries, we also have druggable genome and the whole genome. And here is the list of all the 19 different libraries that we have in our catalog. And this particular column shows the different number of genes that are covered under each sub-library and the corresponding number of guide RNA. So essentially it will be 4 times the number of genes. This particular slide shows the vector construct design for both the lenti-Cas9 and the lenti guide RNA. On the right-hand side here, this particular design for a lenti-Cas9 is, as you can see, the Cas9 is driven by EF1 alpha promoter and is also linked to the plastocidin marker using self-cleaving 2A peptide. And the lenti guide RNA in here, the guide RNA as such is driven by the U6 promoter, and it also carries a selection marker, puromycin, that's driven by EF1 alpha promoter. And I would like to point out one thing in here. For the lenti guide RNA, we have not intentionally used any fluorescent markers on here, just so that when people use it for HCS assays, you don't lose one channel during detection. And to address that lack of fluorescent marker in the guide RNA, we also provide controls, specifically the HPRT and the scrambled, basically a negative control, that have the GSP marker on there, and you can use this for detecting transduction efficiency, and also as negative controls in the assay. This particular slide further analyzes how efficient these lentiviral preps are, and Sanjay already went over how the efficiency is detected via GCD, and in this case the same thing is depicted. So basically HT1080 cells were transduced with some of these example proteins that are shown here, and this was dual particle transduction. So Cas9 and lenti guides were co-transduced and puromycin selected for 5 days, and the GCD assay was run on these samples. And you can see for all the targets we get more than 80% indel formation or gene knockout. This next slide gives an overview of CRISPR screening. So in terms of screening, you can either use dual particle transduction like Cas9 and the guide RNA co-transduce and look at the effect, or alternatively, if you have a bigger screen, you can always generate a Cas9 stable line, and then just use guide RNA for transduction. And depending on if you use pool or clonal selection, it can range anywhere from 6 to 12 weeks. The next milestone then is transfection and assay optimization, and this is one of the crucial pieces of screening, because unless you confirm that your assay is working fine, all your conditions you're using are fine, sometimes, like if you just move forward with screening, it might not yield useful data. So the transfection and assay optimization is a key step in any screening process. So once you have that optimized, which takes about roughly about 3 weeks, and it depends on the assay that you're using and the controls. Once you have that optimized, then you move on to the actual library screening, followed by validation, and depending on the library size and the assay format, again, these can take anywhere from 2 to 5 weeks. And at Thermo Fisher here, we offer this as a service. Next, I'll talk about a case study wherein we used the Invitrogen lenti array library, the Kinome library, for looking at NF-kappa B signaling pathway. And for this, we used one of our cell sensor assays in ME180 cell background, and shown here on the left-hand side is the simplistic view of the NF-kappa B activation in this cell line. Upon activation via TNF-alpha, the TNF-alpha would, upon activation of TNFR1 receptor via TNF, you get downstream activation of TRAD, IKK-alpha, further leading to activation of NF-kappa beta, kappa B, which then translocates to the nucleus and activates a beta-lactamase gene. So the cell sensor line expresses a beta-lactamase reporter under control of NF-kappa B response promoter. So once NF-kappa B is activated, it translocates to the nucleus and activates beta-lactamase. And the way we detect this is using a live blazer substrate. And when the cells are loaded with live blazer substrate, if there is no beta-lactamase production, the live blazer substrate is intact and it basically works on FRET-based mechanism. So when you excite the substrate at 409, it emits in the green range. So essentially green cells means no activation of the pathway, but when beta-lactamase is present and due to activation of the pathway, it cleaves the beta-lactam ring in this live blazer substrate, and because the ring is cleaved, when you activate the substrate at 409, it emits in the blue range. So essentially blue cells means the pathway is active. So just in the next few slides, you should remember that green is pathway is not activated, and blue is pathway is activated. And we look at the blue-green ratio for further analysis. And this particular slide summarizes how we go about optimizing the assay for downstream screening. The top panel here shows how different cell feeding densities compare to using different MOIs. So we used 1,000 cells per well or 2,000 cells per well and 4 different MOIs, and you can see that MOI of 2, which is shown by the blue bar here, is the optimal, because using higher MOI does not provide any additional advantage. So to save on the reagents, then MOI is like the optimal one to go with. And the next condition that we looked at was spinoculation. This is like when we transduce the cells or overlay the cells with lentivirus and spin it at high speeds of like 2500 for 30 minutes. So in certain cell types, this is shown to be useful. So we wanted to see if spinoculation is needed in this case or not. And when we compared, spinoculation is shown by the red bars here. And when you compare it to no spinoculation shown by the blue bar in the middle plot, you can see that it was very comparable. And we also compared like different days, reading at day 5 and day 8. So essentially there was no difference between day 5 and day 8, and you can have an early readout of this assay. And spinoculation, as you can see, the blue bar and the red bars compare really well, so it did not have any additional benefit. So in this case, it's optional. Similarly, with puromycin selection, it depends on the transduction efficiency. If your transduction efficiency is really good, you do not need any antibiotic selection, which is shown in this case. Like the red bar is wherein puromycin was, the cells are selected with puromycin. The blue bar is shows where no selection was added to the cells. And again, in this case, they do compare very well. That means the transduction efficiency in this case is so high that you don't really need to add selection. And then again, reading at day 5, day 9 really did not make any difference. That means a shorter time frame for assay is just fine. So having optimized all these conditions, we then further validated the known targets in this NF-kappa B pathway, and followed the protocol that's listed here in the workflow on the top, wherein we take the we take the clone that we generated that stably expresses Cas9. We seed cells, that is day 1, and on day 2 we transduce it with the guide RNA. And 4 days after transduction, on day 5, we add TNF alpha stem and read the blue-green ratio. And shown below here are the images from the control samples, and you can see with scrambled and HPRT, which are negative control, you see that the pathway is active. And when we treat the cells with guide RNA targeting BLA, which is like a technical control, if you knock out the reporter, you should not see any signaling or activation of the pathway. So we indeed don't see any activation of the pathway in this BLA slide. And the same thing is true when we knock out TNFR1, you do not see activation of the pathway, because if the receptor itself is knocked out, you should not ideally see any downstream effect. And then IKK alpha and TRAD, which are the partials, now we see a little bit of activation, but mostly the pathway is inhibited. So having confirmed this, we then went on to further validate if what we are seeing in the functional assay, does it correlate to what is happening at the genome level. And here you can see in the top plot, we have shown TRAD treated cells and IKK alpha knocked out cells. And the indel formation was determined by NGS assay, and you can see it's really very highly efficient indel formation, more than 80% in both cases. And when we looked at the protein level, in fact, in these cells, as shown in the lower plot, you do see good knockout of these both proteins. And then we looked at the functional assay. The indel formation really translates well to the functional assay as well, wherein we see more than 85% inhibition of the pathway or beta-lactamase activation, and the images on the top show the same thing. This is a secondary verification. Say if somebody is, if you're not interested in looking at the beta-lactamase pathway or beta-lactamase reporter, you can also use this system to study via HCS or imaging system. And on the right-hand side plot here, right-hand side panel images, what you are seeing is NF-kappa B translocation to the nucleus upon TNF alpha activation that's shown in the lower half of the left-hand side panel. And in the cells that are TRAD knocked out or IKK alpha knocked out, you do not see this NF-kappa B translocation to the nucleus, indicating that the pathway is disrupted. On the right-hand side panel, we looked at the IKK alpha IKK alpha activation using anti-phosphorylated IKK alpha antibody, and you can see in the parent or the no guide RNA treated cells, you do see IKK alpha phosphorylation upon TNF alpha treatment, whereas in the IKK alpha knockout cells, you do not see that activation upon TNF alpha stimulation. So next, after having thoroughly validated the system, we went on to the actual screen, wherein we used 384 well format for screening and the other parameters that we had previously confirmed during optimization, and the controls that are listed in here, basically HPRT a negative control, BLA as a technical control, and the pathway control, CFOS, IKK alpha, and TNFR1. And for this, we screened the whole kinome library that consists of 840 targets. And we used the same assay protocol that I previously described, which is a 5-day protocol. And this was the plate layout that we used. So we screened about 56 targets on each single 384 well plate along with the controls. So the 10.5 plates for the kinome library, the 10.5 96 well plates of the kinome library then translates to 15 384 well plates. And the data from the controls from these 384 well plates is shown here on this plot, which looks at the interplate reproducibility, and you can see that there are 15 dots for each of the plots, and it is really very tight, showing that this assay is robust and works really well for each of the, especially for the controls, TRAT, TNFR1, BLA. And this is what the actual final screening data looks like. So plotted here on the y-axis is % activation of the pathway, and the x-axis is the number of samples. And shown here on this particular right-hand side plot is the same controls. What you see on the left-hand side are just as a reference point, and depending on the reference point, then we drew that arbitrary red line which separates the true negatives from the positive hits. And as you can see, some of the colored dots that are shown on the plot, like the PLK4, CHOC, NEC4, KN1, are the known, very well characterized players in the NF-kappa B pathway. So we did identify already known, and along with some of the unique targets. So having identified 75 or so hits, it ranged from cell surface molecules to molecules involved in cell cycle regulation. We did confirm some of the other hits using small molecule. Wherever we could find a small molecule available for a particular target, we validated that, and that data is shown in this plot here on the right-hand side, lower right-hand side, wherein we confirmed the PKN1, PLK4, and some other targets. We did confirm that it was indeed a target for this particular pathway. Next, we went on to further validate these hits using siRNA, and shown here in this plot on y-axis is the BLA pathway inhibition, and on the x-axis it's the knockdown of the particular target gene using BacMam gene expression assays. And you can see using these siRNA, we got pretty good knockdown for most of the targets. And in some cases, the effect on the BLA inhibition was really good, but in for some targets, it was not that impressive. Then we compared this data to what it looks like with CRISPR, and shown here, on the right-hand side plot, on y-axis is siRNA-mediated inhibition, and on x-axis is the CRISPR-mediated inhibition. And you can see for some of these targets that are on the extreme right-hand side of the plot are some of the very well-characterized molecules or targets in the pathway. So no wonder they show up in both CRISPR and siRNA-mediated. Whereas some of the other ones that are really subtle or do not show up in siRNA, the one in around that 50% mark around in the CRISPR screening, are really, really low in case of siRNA-mediated inhibition. So these could have been easily missed in a siRNA screen, but the CRISPR screen picks it up, indicating that the CRISPR knockout is more penetrant as compared to the siRNA knockdown. Next, quickly, I just want to talk about how these CRISPR libraries were used to identify a new therapeutic target in case of rhabdoid tumors. One of our collaborators, Dr. Simone Shredney at Northwestern, she did this study, and using our Lenti-CRISPR library, she identified PLK4 as a target for the malignant rhabdoid tumors, and this work was published earlier this year. Just the gist of data from the study, the middle plot here shows one of the results from Simone screening, and what you see here is the cells that were PLK4 knocked out showed the slowest cell growth as compared to any other targets that she looked at. This indicated that the cell growth was inhibited if PLK4 was knocked out, and we further confirmed that in these cells where PLK4 was knocked out, in fact you see on the extreme right hand side you see good indel formation, indicating that the gene shows very high efficiency of knockout. And she further confirmed the slow growth rate using Ki67 or cell proliferation marker, and you do see significant difference between the cells wherein the PLK4 was knocked out. It was significantly, the proliferation rate was significantly different as compared to the parental cell line or just Cas9 expressing MON cells. And on the extreme right-hand side, it is a clonogenic assay wherein you look at clone formation. So what you see here on the left-hand side plate is just the parental cells, MON Cas9 cells, and on the right-hand side is MON cells wherein the PLK4 is knocked out, and you see very few colonies formed there. So one of the interesting thing is why wasn't PLK4 picked out in this case prior to Simone's analysis, and this particular plot here alludes to that. So if you look at PLK4 expression in the rhabdoid tumors or the atypical teratoid rhabdoid tumors, it is the PLK4 expression is not very high or is not significantly higher as compared to other embryonal brain tumors, but it is significantly high as compared to the non-embryonal brain tumors. So it's easy for it to have been missed, but because using CRISPR and total knockout of the gene then helps to identify that as a target. So what Simone did in this case is she collaborated with one of the group up in Toronto, Canada, wherein they had already identified a PLK4 inhibitor, the CFI40094, as one of the molecule for breast cancer treatment, and it was already in clinical trials. So she used this to see what effect it has on the HPRT tumors, and shown here on the right hand side in the clonogenic assay, then the compound treated cells do show inhibition of growth or no colony formation, and that was true across other cell lines like BT12, G401 or BT16, the other HPRT cell lines. So it has the CFI molecule has a true potential of being used as a drug for treatment of this HPRT tumors. So in summary, at Thermo Fisher Scientific, we offer full workflow solutions for editing and functional genomic needs. The CRISPR libraries, as I talked about, are powerful and high throughput loss of function screening tools for target identification. And although we offer like the 19 libraries that are listed in our catalog, the CRISPR libraries are built in such a fashion that they are flexible, and if needed, we can make a custom library as per your needs. So we can put together a custom library if needed. So essentially, yeah, we have all the tools that you need for your genome editing and screening efforts, and make a big impact on drug discovery and your screening efforts. Last but not the least, I would like to remind you that we have another event on November 9th. It's the 24 hours of stem cell. So please feel free to register for this event. And it's on November 9th, and the registration is free. And thank you for your time and attention.
Thank you so much, Chetana and Sanjay, for that informative presentation. I now want to turn to our audience for the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on that green Q&A button at the lower left of the presentation window. Type your questions into the box that appear on the screen and click that send button. We'll answer as many questions as we have time for today. So let's take a look at our audience questions that are already coming in. Our first question is directed to you, Chetana. What's the advantage of using lentiviral vectors compared to plasmids and AAV?
So the key advantage of using lentiviral system is mainly in cell types that are hard to transfect or transduce. So. And then even primary cells or non-dividing cells. So essentially it can be used across a broad range of cell types. That's what I see as a key advantage. And the lower MOIs, most of the cell types you can get away with using lower MOIs or multiplicity of infection that you use for transduction.
Wonderful, thank you. And here's another question actually for you as well. What does ATG5 do?
Oh, ATG5 is one of the key components that's involved in autophagy. So it is involved in, it is mainly, the primary role is it's in the autophagosome formation.
Wonderful,
so it's one of the core proteins.
Okay, thank you. And thank you again, audience. We have great questions coming in from all over the world. Here's our next question, and this one's for you, Sanjay. You spoke about a lot of different methods to detect the edit. What methods do you find that are best or used the most often, and why?
Actually, we use most of the methods. It's just that there are certain methods which are easy to pursue to ask simple questions. For example, I showed the gel-based genomic cleavage detection method. That is a very easy method to ask very preliminary questions like, okay, if I designed more than one editing agent, let's say we designed 3 CRISPRs, which one worked best? But that particular question can be very quickly answered by genomic cleavage detection assay. But if we are asking more detailed questions, for example, whether the intended change that we wanted to introduce into the cells was introduced and what was the frequency of introduction of that change, that question will be answered by sequencing. So there are different places where both of these different methods are applicable. One is for simple asking a yes or no question, and the second will be answering some more detailed and in-depth questions.
Wonderful. Thank you so much, Sanjay. And here is another question for you. Excuse me, I'm sorry. Let me pull it up here. I apologize. It is all of a sudden not showing. The next question is actually for you, Chetana. How will you apply this study a Mendelian disease like cystic fibrosis?
Oh, that's a tough one. Huh.
So actually it's a interesting question, right? We can actually address things like cystic fibrosis. Cystic fibrosis, one of the key determinant of cystic fibrosis is a mutation, a frameshift mutation in CFTR gene. So we can in fact take wild type cells, design the appropriate CRISPRs and appropriate donor DNAs, and create the mutation in the wild type CFTR gene and ask the relevant biological question. Depending on the tissue where it's derived from and the mutation, the zygosity of mutation that's introduced, we can in fact address and ask the relevant biological question in this context.
Thank you so much. And here's one more question that we have time for today. And again, I want to thank our audience for their participation. Our next question is directed to you, Sanjay. What has been your success rate for genome editing? Are there any cell lines that are more challenging or difficult to edit?
It actually is always a mixed bag. In terms of success, we have had a very high degree of success in our genome editing experiments. However, success is again defined by the endpoint, whether we were able to obtain the desired result. However, that said, there are cell lines which are difficult to edit, and there are cell lines which are derived from blood as well as brain. They tend to be difficult to deliver, and so delivery of editing agent, if that is compromised, of course the final end result will be that there will be low level of editing. Now that said, it will only tell us that the amount of editing events within those cellular populations is low. Again, that informs us that the number of clones that we will have to screen for final outcomes will be correspondingly larger. So when we are talking about understanding the nature and frequency of editing events within a pool, the reason why we do that at that level and in that much depth is to understand what is the total number of editing events happening in the total cellular population. And that informs us how many clones we should be screening. Once we have got that number, we can screen the appropriate number of clones to achieve the final outcome. So answer is yes, we can in fact get the outcome. It's only the amount of labor that goes into getting to the final outcome that changes. Hope that answers the question.
Thank you, Sanjay. And Chetana and Sanjay, thank you so much for your presentation today. Audience members, we appreciate your live participation, and any of your other questions will be answered via email by Thermo Fisher Scientific. I'd also like to thank Thermo Fisher Scientific for sponsoring today's educational webcast and LabRoots for making it possible. For more information, you can visit our sponsor at thermofisher.com. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through April of 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thank you so much for joining us. Thank you, Chetana. Thank you, Sanjay. We hope to see everyone again soon. Goodbye.
Bye-bye.